Chapter 7 is nutrition, ecology, and growth of the microbes. And this is just an introduction, nothing so important. How the microbes get their nutrients. They can get it from anything around them. Whatever is available in the environment, and we will talk about this. This is just an introduction. They take the chemical substances that they need from their environment somehow, and we will discuss the details. What drives the microbial engine? They basically require some energy somehow. What the way they will get it, whatever it is, they need energy anyway. So this is what's driving them. I need energy for senses, for growth, for transport, for all these activities. I need energy. How to get it? We will discuss it. And the way they get these chemical compounds, it has to be transported through their membrane somehow. Something outside, they transport it into the inside using different types of mechanisms that we are going to discuss, but it's not free moving anyway. This is the idea. Nothing is freely moving, nothing like enter or leave. No, they decide what should go in, what should go out, and they decide how exactly to transport it, which is the transport machine that we will discuss. Beside that, they need to adapt to the habitat around them. So they live in this environment. If it is dark, for example, if they don't have enough sunlight, they are going to change their color, like this type of pink algae. They basically absorb light instead of reflecting. Other colors are going to reflect. They absorb. Why? Because they need the energy anyway. They need the sun energy. They need photosynthesis. So they need to absorb as much as possible. They are going to adapt themselves somehow. And we have a lot of details to talk about, but generally speaking, to know, it's just they're going to adapt somehow. They do some sort of partnership. Some organisms, microorganisms, they live beside each other, and there is a relationship between each of these organisms. If they live beside each other, they can be beneficial to each other, like I benefit you, you benefit me, or one can benefit from the other without harming them, or they can harm them. There are different ways that they survive together. The relationship is different. It can be whatever, cooperation, or one is using the other, and so on. But there is a lot of partnership and relationships that we will discuss. The way each one of these microorganisms expand their population. So you have this number. If you leave it alone, it's going to die and it will be eliminated. That does not happen because of they expand their population by producing offsprings. They reproduce basically. They have life cycle. They do division. They reproduce in order to expand their population. So part of them will die, but the other part will continue to expand. Nutrients. They are going to need, microbes in general, they are going to need two types of nutrients. One type of nutrient that they need a lot of it, and one type that they need a little bit of it. And these are called essential elements. Generally speaking, if you say bioelements, what do you mean? These are the basic requirements of life. This is what I need for life. If you don't have that, they are not going to survive. Generally speaking, what's bioelements? Bio means life. Elements, the elements that are needed for life, required for life. What is it? Carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, calcium, all of these. This is all the basic requirements. So we call those the bioelements. Essential elements, or the essential nutrients, I mean, this is the substrate that you will need as a source. And as I mentioned, some of them you will need them in big amounts. And I call that macronutrients. What macro means? Macro. A lot of it. So macronutrients, this is what I need, a lot of it. Large quantities of these nutrients. And these are the organic molecules, basically. Proteins, carbohydrates, fats, nucleic acids, stuff like that. The organisms need a lot of those for metabolism. At the same time, they need a little bit, which is called the micronutrients. Micro means small, right? Very tiny. Trace elements. You call it also the trace elements. Micronutrients or trace elements. It's the same thing. So I need just a little bit of other things, small quantities. And these, I need a little bit of it, mostly for the enzyme function. Like magnesium, like zinc. What do you need magnesium and zinc? Is this to build your body? No. What's needed to build the body of these organisms are the macronutrients. Why do you need the trace elements then? It's just needed for the enzyme activities. Like zinc, for example. Some enzymes will not work if you don't have zinc. It's not a basic unit. You don't build using it, right? It's not like a brick, but it's needed for the enzyme function. Some of them will be needed just to maintain the protein structure. Like, do you guys remember from A&P, you get amino acids and then you put them together to make protein somehow, and you fold it. Just to keep it folded like that, you need some trace elements. Magnesium, zinc would be an example. So what's the difference so far? Macro elements, I need it in large quantities. These are the organic molecules, carbohydrate, fat, protein, nucleic acid, right? I need it for the basic building of the cell. I need a little bit, small quantities of the trace elements. This is just for the enzyme function and to keep the structure of the protein. When you say organic nutrients, like I mentioned carbohydrates, fats, protein, and nucleic acid, what do you mean by organic nutrients? When you say the word organic, you mean they have carbon and hydrogen combined. Not carbon alone, not hydrogen alone. It's something, and it's a molecule that have both and carbon and hydrogen at the same time. Examples: carbohydrates, fat, protein, and nucleic acids. This is A&P, right? Remember this? Carbohydrates, fat, protein, nucleic acid. These are the four elements that we all know. But for the organisms, methane can be another thing. They can produce it, and something else can use it, so they can produce methane. They can use carbohydrates, fats, proteins. Fat is the same as lipid, of course. Protein, nucleic acid, and these elements can be used by another organisms. So they can produce methane, carbohydrates, fat, protein, nucleic acid. So this is all called organic. So what's organic? What do you have? Carbon and hydrogen in the same molecule, right? Inorganic is you don't have both of those, just the other way around. You don't have both of them combined. So if I asked you like carbon dioxide, is that organic or inorganic? It's inorganic, right? But it has carbon. Yes, no hydrogen. You have to have both of them at the same time. Water, what do you think about water? Is that organic or inorganic? Huh? It's inorganic. You have hydrogen, yes, but no carbon, right? So it's inorganic. You have to have both of them. Metals and salts are considered inorganic. Anything that does not contain carbon and hydrogen combined, we call that inorganic. So organic, your clue is carbon and hydrogen combined. Okay? Anything else, it's not. Magnesium, phosphate, gases, oxygen, carbon dioxide, water, all of these are inorganic. If you look at this cell of the microorganism or the organism in general, you will find out that 70% of it is water. Do you remember the human cell? Most of it was water too, right? It's water. This is the vast majority of it. And if you're talking about the weight itself, beside the water, what kind of compounds do you have? The vast majority are organic compounds. Remember when we said organic and inorganic? Organic molecules, this is the main thing. This is the basic structure, right? The basic structure is organic compounds. What are the organic compounds that you know? Or organic compounds examples. Proteins, sugars or carbohydrates, fat, nucleic acid, right? These are all organic molecules, but the vast majority is proteins. Like if you look at the cell, you will see that the cytoplasm is mainly protein. The membrane will have a lot of protein, so it's protein. The main thing is protein. If you put together the elements that make the major part, like you say water. What's water? It's hydrogen and oxygen, isn't it? Organic molecules. What do you mean organic molecules? I mean carbon, hydrogen, oxygen, plus or minus other things, nitrogen, phosphorus, and so on. You can remember it, schnapps, CHNOPS, schnapps. CH, C carbon, H hydrogen, N nitrogen, O oxygen, P phosphorus, S sulfur. These are the compounds. If you break down what you come to at the end, give me that protein. What do I have in the protein? It's carbon, hydrogen, oxygen, and nitrogen, and so on. Give me that ATP. Give me that nucleic acids. Tell me what do I have. You have carbon, hydrogen, oxygen, and sulfur, or phosphorus, for example, and so on. So you have to have. It will come up at the end that you have to have these together somehow, parts or all. How about the nutrients? What do they need? I need the 6 that make my own body, right? Which is schnapps, plus other elements, something like sodium, potassium, calcium, magnesium, and so on. What's the source of nutrients that I need? Well, this depends on the organism. Which organism are you talking about? Is that an organism that can take the nutrients from the air? Does it live in the water? You need water or feed on the water. Does it live on other things, living organisms? Some of them even live on volcanoes, and they can take their nutrients from there. They can take it from the soil. Some of them lives on the rocks. Some of them even lives on the ocean water, and so on. So they can get it, you can call it anywhere on earth. How do they get nutrients? Anywhere on earth. Anything, right? Rocks, even rocks, volcanoes, air, water, rocks, salt lakes, even salt lakes. You think there is no life there? No, there are some that they can take their nutrients from there. So simply, everything. Everything on earth. You always find something that the organisms can survive on. Now, how can you get the nutrients that you need? This is very important to understand. Remember, I need 2 things. Okay? You need to give me, in order to get my nutrients, I need 2 things. You need to give me a source of carbon, a source of energy. Okay? A source of carbon, a source of energy. Beside the other things that's available anyway. Okay? Like, you get the other things. Water is everywhere. You get, or hydrogen, you get the hydrogen from here. But the main things, the main bulk, you have, this is how we classify. Do I need anything else? I do. But I'm talking about how to classify. Are we following? In order to classify, tell me, where did you get the carbon from? And where did you get the energy from? Is that clear so far? So let's start with carbon. Tell me, how did you get your carbon? Did you get your carbon on your own, by yourself? I call that autotroph. Auto means self. Troph means feed. So autotroph is you feed yourself. So I don't need any other organism to survive? No, you don't need it. You feed yourself. Clear so far? I call you autotroph. You feed yourself. And the source of carbon in this case is carbon dioxide. carbon dioxide. So do I depend on something else to survive? No. Carbon dioxide, who do I need to get the carbon dioxide? I don't need anything, right? Carbon dioxide is in the atmosphere. Why do I need anything else? Plants, for example, some algae. Plants need any other organism to live? Like, do I need animals? Do I need humans? No, just leave them alone and they will survive, right? What's the source of carbon? Carbon dioxide. Isn't that all over the atmosphere, in the environment? So that's autotroph. On the other hand, and this is what we call the producer, because the autotroph, they feed themselves and then we use what they built, right? The plants, they feed themselves. The source of is carbon dioxide that's already in the atmosphere. They don't need anything else. These are autotrophs. And we call it the producer because we consume them, right? Humans consume them. Animals consume them. Fungus consume them, right? You consume it. So the consumers, on the other hand, are the heterotroph. Hetero means other or different. What's troph means? Feed. So heterotroph, you feed on something else, right? Which is, again, come up to the carbon source. What's your carbon source? I cannot get my own carbon source. I need another organism to get my carbon source. So what's that? Where do you get it from? From other organic molecules. What are the organic molecules that I mentioned several times? Carbohydrates, fats, protein, nucleic acid. Carbohydrates, fats, protein, nucleic acid. Nucleic acid. Is that clear? Carbohydrate, fat, protein, nucleic acid. All of these, didn't we say in the beginning that organic molecules contain carbon and hydrogen? So that will be your source. So you need to get it from somewhere else. Like, are we as humans, are we autotroph? You're not autotroph. Can you survive like getting carbon dioxide from the air? Of course not, right? But what you need to survive, you need to give me another organism, right? You give me plants to eat, you give me animals to eat. From these plants and animals, I get the organic molecules and I can feed myself. Animal, if you don't have plants on earth, all animals will die, right? Because they are heterotroph. They need a source of carbon. Where do you get the source of carbon? From something else. Same for the fungus. The fungus, they need to feed on something else. So here is a question. How do you classify autotroph versus heterotroph? Source of what? Carbon. Autotroph, heterotroph, depend on, classified according to carbon source. Can you get your own carbon from carbon dioxide? You're autotroph. If you cannot, it's heterotroph. Okay? The other way to classify is what's your source of energy. If your source of energy is photosynthesis, which is you get it from the sunlight, right? I call you phototroph. What's troph means? Feed. Photo means the light. Photo, right? So phototroph, you feed on light. Photosynthesis. Sun. What's the example of that? Plants, right? Or, no, I cannot, like us, like humans, like animals. You cannot do it using the sunlight. I'm talking about the energy source now. So how do you get it? Again, from chemical compounds, right? From chemical compounds. We call that chemotroph. Are you getting your energy from sunlight? Phototroph. Are you getting your energy from molecules, from chemical compounds? You call it chemotroph. Is that clear so far? And now you can do a combination, right? You can combine these together. So you can be autotroph, heterotroph, depending on what? Source of carbon. Autotroph, heterotroph. What about your source of energy? Photo or chemo? Phototroph, chemotroph. So you can say photoautotroph. like plants, right? Chemo-autotroph, chemo-heterotroph, right? You can do some combinations. Okay, can you answer this question? If you understood what we talked about. This is two parts. Remember, source of carbon, auto or hetero, right? So it's saying you need large organic molecules to get your carbon, so this is one, and to get your energy, both of them. Which one? Is that, okay, let's take it part by part. Is that photo or chemo? This is chemo. Is that auto or hetero? Huh? Hetero, so which one? C. Yes. Did you get the idea how to do the combination? Just divide it into two parts. Show me the source of carbon, show me the source of energy. Okay? So if we're talking about the autotrophs, what do you mean by autotrophs? Self-feeders regarding what? Carbon or energy? Carbon, okay. So we're talking about autotrophs now. Photoautotrophs. Translation. What's the source of carbon? Carbon dioxide. Self. Photo. What's the source of energy? Light. Photosynthesis. Okay, so now these photoautotrophs. Usually when we say photoautotrophs, we're talking about mainly plants, right? But not only plants. Not only plants that do that. And these photoautotrophs, including plants and algae, and some types of bacteria. So remember, it's not only plants that are autotrophs. We have some bacteria that are autotrophs. They don't need us. Yes, they survive on their own. They are autotrophs, photoautotrophs. Bacteria, yes. Plants, algae, bacteria. But we are going to subdivide those into two types. Tell me if you produce oxygen or not. Okay, you are photoautotroph, I understand that. But what do you end up producing? If you end up producing oxygen, I call you oxygenic phototrophs or photosynthesis. If you do not produce oxygen, I call you oxygenic photosynthesis. What's an mean? No or not or without, right? So you are photoautotroph, okay, but tell me what you produce at the end. Oxygen or not? If you produce oxygen, I call it oxygenic. Plants are oxygenic. Algae are oxygenic. Cyanobacteria, this is a different a type of bacteria that is photo, so it uses the sunlight, right? And it's auto, they feed themselves, they get carbon dioxide as the source of carbon, and they end up producing oxygen. The other type is the same, but they do not produce oxygen. And instead, they produce sulfur. And the example of that is purple and green sulfur bacteria. This type of bacteria, they are autotrophs, they are phototrophs, but they end up producing sulfur, not oxygen. Okay? Purple and green sulfur bacteria is a good example that you need to remember. Okay? And instead of the chlorophyll, like in plants and algae and cyanobacteria, they have something called chlorophyll. But the most important thing to remember here is these are phototrophs. Photoautotrophs, okay? Auto, you get your carbon from carbon dioxide. Photo, you get your energy from photosynthesis, which is sun, right? What do you produce? This is a subclassification. Oxygenic and anoxygenic. What do you mean anoxygenic? You don't produce oxygen at the end? Yes, I produce sulfur. Like purple and green types like this, or purple and green, different types. Um, so we discussed photoautotrophs. Is that okay so far? Photoautotrophs. How about the chemoautotroph? Like for chemoautotrophs, divided into two parts. Because this is important, and that's why I'm focusing on it. Okay? What do you mean by autotrophs? This is a source of what? Carbon. Of carbon. So, do you need something else, or you can feed yourself from the carbon dioxide available? Auto. You feed yourself from the carbon dioxide. Okay, this is the first part. Auto. Chemo is a source of what? Energy. So if it is chemo, is your source sunlight or another chemical compound? Another chemical compound. Okay. So chemoautotroph, they can feed themselves from inorganic substances around them. And a good example of that is methanogen. What's gen means at the end of any word? Generation. To generate. Methanogen are the methane generators. They generate methane. This is a chemoautotroph that produces methane at the end. Are we following so far? Remember the autotrophs, we subdivide them according to do you produce oxygen or not. If you don't produce oxygen, like you're producing sulfur, for example, okay? How about the chemotroph? Tell me, what do you produce at the end? If you produce methane at the end, I call you methanogen. Okay? And if that's the case, if these methanogens, in order to make the methane, they need a source of hydrogen, carbon dioxide, and it has to be anaerobe. What's anaerobe means? Without oxygen. I have to survive in the absence of oxygen. Okay? Don't give me oxygen. So they do not need oxygen. What do you need to make methane? Hydrogen and carbon dioxide. Where can you get it from? From the environment. Why did I say environment? Because it's O2. Isn't it? Okay. How about the heterotroph? Heterotroph, chemoheterotroph, what's hetero mean? Referring to the source of what? Carbon. Do you get it on your own, or you need to get from another molecules from another organism? Another organism. This is the first part. And chemo means this is the source of energy. Do you get it from sun, or do you get it from also organic molecule, or from a molecule, a chemical compound? Where did you get the source of energy from? Chemo. From a compound. Okay, so they get the carbon and energy, both of them, from organic molecules. Like those who do aerobic respiration. We do aerobic. What aerobic respiration means? What's aerobic? Oxygen. Like us, like our cells. Our cells get the oxygen and they use it to burn glucose, for example. Do we remember this from A&P? You get the glucose into the cells, and then you get oxygen into the cells. You burn the glucose using the oxygen. This is anaerobic, and this is chemoheterotroph. We are example of chemoheterotroph, okay? Humans are example of that. I depend on organic molecules in both sources, carbon and energy. Is that clear so far? Okay. Now if we're talking about organisms, these organisms, the subdivision of the chemoheterotroph. So chemoheterotroph regarding organisms. Your source of energy and your source of carbon, both of them, is from organic molecules. Now, since you are hetero, you mean you are using another another organism, right? What is that organism? It depends. If you are using dead organisms, I call you the saprobes. Saprobes, the decomposers, the saprobes, right? If you are if you are using a living organism, a host, I call you parasites. Is that clear? Are you a saprobe or parasite? Saprobe means, generally speaking, saprobe means you live on dead organisms. Parasite, you get it from the host, but here's your clue for parasite: you harm. You harm. You take it from the host and you harm the host. Is that clear? This is your clue here. When you say parasite, meaning not only that you get it from the host, but you harm the host as well. I call that a parasite. So they cause disease, like the parasites that we talked about, like these helminthes, right, that we talked about before, these worms. They do not only infect us, they do not only they take their needs from us, but they harm us, right? So we call it parasite. So what do you call it when you live on dead organisms? Saprobes. And what do you mean by parasite? This is they take their needs from the host and they harmful to the host. Okay? Some organisms will have a cell wall, like bacteria, for example. Okay? If they have the cell wall, what's the significance of the cell wall? Do you remember when we said that in the very beginning, when we discussed the prokaryotes, when we said that the cell wall is there to protect them so they are more aggressive? And this is one of their aggressive criteria. They are more pathogenic. So if they have a cell wall, the cell wall is protective. The cell wall protect them. If you don't have a cell wall, we can kill you, right? So you are weak. If you have a cell wall, you are stronger. So now here's what's happening. If you look at this picture, this is like a bacteria or something, and they are heterotroph. So what they do is they release the red, which is the organisms. They are surrounded by organic molecules, right? Organic molecules. Heterotrophs. They are heterotrophs. The source of energy is organic molecules. How are you going to use these organic molecules? Look at this picture. They have enzymes inside of them, right? These enzymes are allowed to move out through the membrane and the cell wall. Is there a difference between cell membrane and cell wall? There is a difference. What's the difference? Every cell has a membrane. If it doesn't have a membrane, it's not a cell. Is that okay? Yes. All right. Outside of that membrane, you can have a wall or not. So the wall is optional. The membrane is not optional. And the wall is outside of that membrane. Is that okay so far? So these have the enzymes inside of them. The enzyme will leave out. It will go out through the membrane and through the wall also. And they are going to go to these organic debris or organic molecules. They are going to break it down, and then you will take in the simpler forms. Why don't you just take it in? They cannot. Is that clear? Like I'm here, I'm a bacteria or something, and I see outside here organic debris, organic molecules, big organic molecules. I need it. I cannot get it in. What should I do now? I release my enzymes outside, break it down into a simpler form, and then I can take the simpler form. If that's not the case, it's too big. So if you look at this, you degrade them, you break them down into smaller molecules, and then the smaller molecules, you will be able to cross them across the wall and the membrane. Which one is outside? The wall or the membrane? Which one is the outermost? Wall. The wall is outside the membrane. So it will cross the wall, then cross the membrane, then you take them into the cytoplasm and use them. Okay? This is summarizing all what we talked about. Photoautotroph, chemoautotroph, and chemoheterotroph. Okay? And these are examples, so this is a good to summarize. And it has examples that you need to know. Saprobes, like what? Like there are different types of fungi, bacteria, and protozoa. So we have photoautotroph, chemoautotroph, chemoheterotroph, and photoheterotroph. So a combination between both of those, right? Photo heterotroph, the purple and green photosynthetic bacteria. Purple and green bacteria, they are photoheterotroph. So what does that mean? Split it into two parts. What's hetero? They take the carbon source from another organism. This is the first part. Photo. They take the energy from sunlight. Oh, so they do not count on the organic molecule for both? No. This is rare example, but remember it. Purple and green photosynthetic bacteria. Because this is something very rare. Usually if you're hetero, you are chemo, right? Chemohetero. This is the vast majority. You take your source of both carbon and energy from organic molecules, right? This is a rare example. That you take the sources from your source, organic molecules for carbon, but not for energy. Okay? Now the transport. Here you have the example that I gave you, an organism that have these organic molecules outside, and these organic molecules are big. I'm going to break it down and then I will take it in. Clear? How to take it in. This is called the transport. How are you going to transport? So listen to this. This is the main way to classify those. Okay? I'm going to move something inside of me. Alright? It will depend on two things. It can be passive, it can be active. What do you mean by passive? What do you mean by active? I mean by passive, no need for energy. Active, you need energy. Now the question is, passive, why you don't need energy? Because I'm moving something via concentration gradient. Why the active is active? Why do you need energy? To use it for what? You're moving something against concentration gradient. Okay? Did we get the idea? What's passive? You don't need energy. Why you don't need energy? Because you're moving with a concentration gradient. What's active? You need energy. Why? Against concentration gradient. Okay? Like this. This is an organism. I already have a lot of carbohydrates, right? And there's some carbohydrate outside. I still need it. Use energy to get it. Okay? And this will be subdivided into the following. Diffusion. Diffusion is just simple, like that. I have a lot of oxygen outside, let's say, oxygen, okay? And I have less oxygen inside. Oxygen diffused in, that's it. Is that active or passive? A lot of oxygen outside, this oxygen inside. This is passive, right? It's just diffusing out. There is nothing that's needed. No oxygen and nothing else needed. Okay? Osmosis is the diffusion of water. This is as something close to diffusion, but this is specific for water only. Do you remember this? So, can you tell me how the water diffuses? If you have a membrane, does it move from higher concentrated solutions to lower, or from lower concentrated to higher? Low to high. Low to high. From hypo to hyper. Okay? If you forgot it, look at this. I have two solutions here. Okay? This is juice, concentrated juice, and this is diluted juice. It will move from the diluted to the concentrated. The water moves from hypo to hypertonic. Hypotonic to hypertonic. Is that clear? It's unlike what you're thinking. You know why? Because when you say hypertonic and hypotonic, you're not talking about water. I'm not saying the solute. I'm talking about water. Okay? And a good example for osmosis is, we did that in A and P. Remember when we put this red blood cell in a hypertonic solution and a hypotonic solution, or you don't remember? If you get this red blood cells and you put it in hypertonic solution, it will shrink, right? Why? Because the water will move from? Exactly. So it's going to shrink, right? Did it move from hypo to hyper? You put it in hypertonic solution. Hypertonic means more concentrating than the cell. So that's what's going to happen. If I put this red blood cell in hypotonic, it will swell, right? Why? Because water moved from?
Outside into inside the cell.
Exactly. Outside that's hypo to the inside that's hyper, right? And the other example is yourself. If you go to swim in the ocean water, what do you think will happen to you? You are going, it's salty, so your water is going to leave. So you're going to get dehydrated, right? What if you go to swim in fresh water, like lake water, and not to swim, I mean to stay there for some time, or you're using the bathtub and you stay there for hours, you're going to swell. You see your hand is getting like whitish and swelling because the water moved into your hand, right? If you stayed for a long time. So the bottom line is osmosis from hypo to hyper, okay? Facilitated diffusion. So this is not just diffusion. No, it's a diffusion that needs some sort of facilitation. Like, I give you example here, oxygen. Isn't oxygen is too tiny? It's 2 atoms. So it can diffuse. But what if I have something that's glucose, for example? Glucose is not small enough to diffuse. You have to have a carrier to carry it in. It cannot diffuse, it's dead. So is it still diffusion? Yes. Is it still across by concentration gradient, via concentration gradient? Yes. But in this case, I need facilitation. What's facilitation? What do you need? A protein carrier. So what's facilitated diffusion? It's a diffusion that needs facilitation, right? What's facilitation by what? Protein carrier. This is just the diffusion. It's passive, like if you get some ink, for example, or any color, and you put it into the water, right? You will see it concentrated first in certain area. Leave it for some time, it's going to diffuse, and then you see the whole solution becoming the same color, right? What happened here is this random movement of the molecule is going to spread, and it's going to go from concentrated areas to less concentrated until the whole solution is homogeneous, right? This is just a passive diffusion. For water, we talked about it, osmosis. The water moved from hypo to hypertonic. And a good example of that is red blood cells, right? So if you look at this, where did I put the cell here? In hypo or hyper? Hypo. We put it here in hypo. See how it's getting in? And it's swelling, and it ultimately will rupture. If this is a red blood cell, and what do you think about this? You put it in hypertonic. The water is moving out, and you shrink or shrivel. What shrivel means? Shrivel. No, shrivel is the same as shrink. Shrivel, shrink, shrivel, shrink. Okay? So this is what's happening here. Let me tell you something here that's very important to remember. This is going to swell, right? And it's going to rupture, right? This does not happen to bacteria. Even if I put it in hypotonic solution, does it swell? Yes. Does it rupture? No. Why? Cell wall. Okay? So you can swell, but you're not going to rupture. This is only if you have a cell wall, right? Red blood cells, of course, it does not nothing of us have a cell wall. So here is the example again. Put it in hypotonic or hypertonic. What if you put it in isotonic? Nothing. Does it move in or out? No, it does not move at all. This is if it's isotonic. So if you put it in hypotonic solution, it's going to swell and it can rupture, and you call that lysis. What's lysis? Breakdown, generally speaking, breakdown. Okay? So in this case, it's going to burst. Does the bacteria burst? No. Does it swell? It can swell, but does it burst? Why? Cell wall. If you put it in hypertonic, it's going to shrink or shrivel, and the cell can die still because it lost its water. So it can cause plasmolysis, but in another way. The second type of still passive and still diffusion is facilitation. Facilitated diffusion. Diffusion means diffuse from high to low. What's facilitated means? What's facilitated? Carrier protein. Okay. So this carrier protein has two criteria that we need to understand. It's specific and it can be saturated. What do you mean? I mean I am the carrier that is specific for glucose. If you ask me to carry anything else, no. I'm specific. Okay? How did I know that this is a glucose? It's like, do I think about it or something? No, it's very simple like this. Look at this. This is glucose. I'm the carrier of glucose. If it fits in, I know that you are my glucose. I will carry you. But what if it is like this? Doesn't fit in? No. So this means it is specific, right? How do you know? It fits in. This is a square, this is a square, fit in. So this is specific. Saturation means the rate of transport. Example, I have 100 carrier, right? You give me some glucose to carry, 50, I can carry 50. Give me 60, 70, that's okay. You give me 100, that is the maximum, I'm saturated. If you give me 120, no, I'm already saturated. So specificity and saturation. So look at this. Look at this, how this is circular and this is circular. So this is specific for this. Look at this. This specific for this. When it fits in, it will open. This is the green here or the purple here. This is the carrier protein. You have to fit in. The shape will determine. On the other hand, if it is active, what active means? Using or not using? Using energy. Why you need to use energy? You're moving against concentration gradient from low to high. Okay? And this one can also be carrier mediated. Carrier mediated active. So carrier mediated can be active or passive. Yes, both of them need a carrier, but are you across concentration gradient or against concentration gradient? With concentration or against concentration? So the first type is carrier mediated, same way as we mentioned before, but it's going against concentration gradient. The other one is group translocation. You're transporting a molecule, but at the same time, you put something on it and you change the nature. Bulk transport this is not a molecule. This is something that's big, right? If it is big, and you're moving it endo or exo, what's endocytosis mean? Inside, but it's bulk, right? So this is not something diffusing across the wall. No, this is you're breaking down the wall, surrounding it, and move it inside. You call it endocytosis. What if you take a big molecule surrounded by part of the membrane and move it out of the cell? Exocytosis. On the other hand, depending on the nature of what exactly are you transporting. Are you transporting a solution or a liquid or a fluid? You call that panocytosis. Pano is coming from drinking. Okay? If you're transporting something solid, like you're transporting a cell, a molecule, solid, you call that phagocytosis. This is depending on the nature. What's panocytosis means? Pano is coming from drinking. Phago is coming from eating. There is also a transporter protein that's called permease. that's help to pump. This just attached to the solute binding protein. There is a protein. When it attached to it, it will open. And using energy. This is for the active. Like, look at this. This is the solute that you're trying to transport. Okay? It attached to a protein, a transport protein first. And this transport protein will attach itself to this, which is permease. It will open the gates using ATP and you go in. Why did you call it permease? Because it gives permission. Permease permission. How about the group translocation? This is another active. But this is just while you're moving something in, you change its structure. Clear? It's unlike the active transport. The active transport carry a protein. The protein, take it just like this. I take it from here, I push it inside. That's it. I didn't do anything, right? This is different. I take it, I put something on it, I change the nature, I make something while transporting it, and then I move it in or out. Okay? So and if you ask yourself, why are you doing this? Like, why don't you just take it from here to here against concentration gradient? To save some of your energy. So we will say, okay, I'm using energy anyway. While transporting this, why don't I make something on my way? Okay? So this is the group translocation. If you look at this picture here, this is something that you're trying to move in. You get it in, you add something to it, you synthesize something on your way to save your energy. You say to yourself, am I going to use energy anyway to transport it because it's low to high? Yes. Why don't me as well, while transporting it, I do some synthesis? Why don't I make something out of it? Okay? Is that clear? What's endocytosis? You take something big. If it is not big, it's not endocytosis. Oh, is that clear? So endocytosis is not just moving something in. You're moving something large. Okay? Like, you guys can get from that door and enter to this, right? This is diffusion, right? Can I get a big truck into this room? No. What's the only way to get a truck here? You have to break down the wall, right? You don't have any other options. Breaking down the wall meaning breaking down the membrane, right? This is only if it is something large. And phagocytosis means eat. Panocytosis means drink. How do you eat and how do you drink? Look at this picture. You're eating using pseudopods. What's pseudo and what's pod? Pseudo means false. Pod means? Yes. So you create like a false foot or feet, surround it and get it done. This is how, if it is eating, if it's solid. What if it is not solid? If it is fluid? You just use the microbial light to push it in. Can you answer this? You have two components here. Which one is it? B. Why? Because it's a diffusion, but you need a carrier protein, right? Okay. Now the environmental factors. The niche means all the adaptations that the organism can do to survive. Temperature can be different. Oxygen requirement, pH, osmotic pressure, barometric pressure. So there are different criteria, and organisms can live in any one of these that's different from one to one. So the first one is temperature. Temperature adaptation. For each organism, there is an optimum temperature, the perfect temperature. And if you give me that perfect temperature, I'm going to grow and metabolize fast. This is perfect for me, okay? What if it is not? Well, I can still work up to a limit, okay, which is the minimum and the maximum temperature. Yes. Example of that, I'll give you an example. If you have flu, for example, flu love your own temperature. They love the optimal temperature is your body temperature. They love it like this. What if you get some fever? Is that perfect for them? No. Up to a limit, they will slow down but can still survive. What if your body is getting colder? They are not going to die. They will survive, but they can take it. This is my perfect, 37. What if it's getting 40? I slow down, but this is the maximum. What if it's getting 33, 35 for some reason? Your body is cold for some reason. Can you still survive? Yes, but this is the minimum. So there is a minimum and maximum and optimum. Optimum is the best for me, fast growth and metabolism. I can go up to a limit, down to a limit. What fails means? Sacrofails. Sacrofails. Don't worry about sacro, just fails. What fails means?
They like it.
Or feel. They like. So here you have sacrofails, which is cryofails. Sacro means cold. Same as cryo. Mesofails, meso, translation, middle. Do you remember mesencephalon, which is the same as the midbrain? So meso means middle. This is, they like, the optimal temperature is the average temperature. Thermophils, they love hot weather. Thermoduric, what's difference between phils and duric? Philic means,
They can endure it. Or they like it. Or they endure it.
phils means they like it. Duric, can you guess?
They endure it.
They can duric, like
For a duration.
it's coming from durable, like it's okay.
Yeah.
I can take it. It's not good, but I can take it. You got the idea?
Yeah.
Okay. So this is very important, by the way. Each one of those definitions is very important. Okay, sacrophils or cryophils, they love low temperature, under 15. Even 0, they are fine with that. Some of these are called the facultative sacrophils. If it goes up a little bit, they can still survive. Facultative. It's not obligated. Mesophil, they like the middle temperature, the medium, the average. Thermophil, they love hot, which is above 45. Thermoduric, they can take it up to a limit. They can survive, but not the best. According to the oxygen requirements, some of them will have to have oxygen to survive. Some of them cannot survive in the presence of oxygen, and some of them can survive in either you have it or not. Do you remember this? Aerobic versus anaerobic? Okay. But the thing is, there is something here that's important. If there is an oxygen in the medium that you're using, okay, you're using oxygen, and there is oxygen. What's the problem with oxygen? The problem with oxygen is it can change into a harmful, toxic product that's called free radicals. Superoxide and peroxide are the most important. So superoxide and peroxide. These are free radicals. And this is toxic. So if the cell have those two enzymes that you need to remember, superoxide dismutase and catalase, if the organism have it, you are good. You are protected. You can deal with the free radicals. I can take care of it. The toxic hyperoxide or superoxide ions, it's a harmful, but it's not harmful to me. I will deal with it. Meaning, if you have superoxide dismutase and catalase, you are protected, okay, from free radicals. If you don't, you cannot survive in the presence of oxygen. So that's what brings us to the aerobic, anaerobic, and facultative. Obligate and facultative. So when you say aerobe, generally speaking, means you can use oxygen, and you do have these two enzymes that can protect you. Okay. Obligate aerobe, they cannot grow without oxygen. What obligate means? They have to. Obligated to. Facultative anaerobe, like E. coli, you have oxygen, you don't have oxygen, it's okay. They can switch their machinery to deal with the situation. Microaerophilic. If you think about the name, it gives it away. Micro means small, a little bit. Aero, air or oxygen. So microaerophile, they love a little bit. They want the oxygen to be small amount. They love it if it's small amount. Okay? Like the soil bacteria. The bacteria in the soil, they love a little bit of oxygen. If you add more oxygen to it, they are going to die. They love it small amounts. Anaerobe, on the other hand, they does not utilize oxygen. They do not utilize it, but can you survive or not? Obligate anaerobe, they cannot survive. What the enzymes that they don't have? Superoxide dismutase, and the other one is catalase. Superoxide dismutase and catalase. You have it, you don't have it. If you have it, you are aerobe. You don't have it, you are anaerobe. Obligate, if you add oxygen, they will die. And by the way, this is used, if you ever heard about concentrated oxygen or hyperbaric. Did you hear about this? Hyperbaric oxygen. It can be used for a treatment of some infections, like some infections that come in the skin, for example. The treatment is hyperbaric oxygen. Why? Because these are anaerobes. Once you give them oxygen, they're going to die. This is the obligate anaerobe. Aerotolerant, the name give it away. Aero means air or oxygen. Tolerant means they can tolerate. The same as diuric. Do you remember diuric? Diuric, tolerant is the same thing, right? If you're making a culture, if this is an anaerobic culture, most of the organisms or microbes, they need carbon dioxide as a source of carbon. Capnophils, capno is a prefix that means carbon dioxide. Okay? Capno means carbon dioxide. Capnophil, they love carbon dioxide. The more carbon dioxide you have, the best for them to grow and metabolize. How about the pH? Same thing. Neutral, acido, and alkalinophils. They love the neutral, they love the acidic, they love the alkaline. Neutrophils, this is the majority. They love the optimal or neutral, which is somewhere around 7, right? Isn't 7 neutral? 7 plus or minus 1, not exactly 7. Okay? This is their optimum. Acidophils, they love the acid, like lactobacillus. Lactobacillus, they produce lactic acid, right? Yogurt bacteria, they also acidophils. They like acidity that you have in the yogurt. Alkalinophils are these different types that they love alkaline. Pseudomonas is an example of that. Yeast, the yeast, they love the alkaline medium. Osmotic pressure. Osmophils means they love high osmotic pressure. What osmotolerant then? They can tolerate the high osmotic pressure. Okay? And osmolarity, if you talk about osmotic pressure, it's mainly from salt. Okay? So if they love salt specifically, you call it halophil. So halophil is the same as osmophil? Not exactly. Halophil is a type of osmophil that loves a lot of salt specifically. Salt is not the only thing that makes the osmotic pressure. Is that clear? So halophil is they love salt specifically. They are osmophils, but they love. So the word, the prefix halo means salt. Okay. So what if this is obligate halophil? You have to have a lot of salt to live. If you don't have it, they are going to die. Okay? Like halobacterium and halococcus. Even if you don't remember the name, what's the first part? Halo. What's halo mean? Salt. Osmotolerant, on the other hand, is they are okay. They can survive salt. Facultative is whether you have it or not, they can survive. Staphylococcus is an example for that. The next component is the pressure. Barophils, they love high pressure. You know that baro is a prefix that means pressure? Okay, so barophils loves high pressure. Like what? Like deep in the ocean, for example. Isn't deep in the ocean is very high pressure? They love it this way. And they can live there and they are fine with that. That will bring us to the one term that's called extremophils. What's extremo means? Extreme. Phils love. They love extreme. Extreme of what? Any of what we talked about. Extreme of anything. Answer this question. Sacrophils? Yes. What sacro means? Cold. Okay. This slide is extremely important to know. Okay? Each single term is important here. Very important. Okay? If you have different microbes or different organisms that live in the same medium, what is the relationship between these organisms? Okay? The relationship is divided into symbiotic and non-symbiotic. Symbiotic means we have to live together. Non-symbiotic means we live separate. Even if we are in the same medium, yes. But you are by yourself, I am by myself. Okay? So symbiotic means we coexist. And this will depend on the relationship between us. Both of us need to be together. Okay? We have to coexist. Coexist in three different ways. We coexist together, I benefit you and you benefit me. This is called mutualism. Okay? If I benefit you but you don't harm me, at least, you call that commensalism. If I benefit you and you harm me, this is called parasite or parasitism. On the other hand, if it is not symbiotic, I am free, you are free, we don't need each other. However, it's okay that you're producing something that I can take it from you. Okay? So this is called synergism, is if you can share nutrients, we do not count on each other. We don't have to coexist. But since you're producing nutrients, I can take it. Okay? We can share. Take some from me, I take some from you. Antagonism is you producing something that destroy me. So this is the same thing, basically, repeating the same thing. It's just adding, synergism is also known as syntropy or syntrophy. Antagonism is the same as amanelism, which is not the most important, but at least remember synergy and antagonism. Synergy, benefit, antagonism, harm. Archaea are not pathogen. If you're talking about all these relationships, what do you classify archaea? Archaea are not pathogens to mammals. Why? Because simply, it's not like they're being nice. We do not have what they need. That's why they do not invade us. We don't have the required environment. Biofilm, we talked about this before in previous chapter. This is a biofilm that form on top of the water. These organisms at certain time, if they don't find what they need, they will float on the surface. They'll stop moving, float on the surface, stick together, stick to the surface, and they are going to affect that water surface. How does it happen? They lose their motility. They make an adhesive matrix to hold them up. They start to form the biofilm that become denser. They enlarge, and they start to become bigger and more and increase their population. And they start to secrete enzymes to digest the surrounding to use it. For human body, the same relationship you can have, it's usually symbiotic. Symbiotic means coexist. The bacteria need you, and some bacteria benefit you, right? So this will be mutualism, right? And so on. So it can be commensals, parasitic, synergetic, and so on. Same thing. So it's basically for humans. If you're studying the microbial growth, you're studying in different levels. How do they grow on a cellular level? How do they increase their size and then increase population? The main way for them to divide is binary fission. Binary fission. This is the main thing. What's the binary fission? It's 5 steps. The first step is when the cells continue to grow, synthesizing what they need, preparing for cell division. Second step is they replicate the chromosomes and the cell become bigger. Third one, they start to make a septum. The chromosome divides, one come to this side and the other come to the other side, making a septum. They complete, now you have 2 compartments that have a septum, and then the end of the cell division when you have 2 daughter cells. If you look at this, this is describing what we talked about. The rate of growth is the rate of completing the time that's needed for completing a whole synthesis. This is called generation time. What's generation time? The time that's needed for a whole generation, meaning to complete a whole fission cycle from 1 to 5. What the time that you need, we call that generation time. There is a curve in which these cells are going to grow, or these organisms are going to grow. It goes with lag phase, exponential, which is the log phase, stationary, and this phase. If you look at this picture here, lag means they are lagging behind. They are not increasing in their population, lagging behind. Okay, so lag means they didn't start to go into a high replication or duplication. Log, which is the exponential part, is when the rate of growth exceeds the rate of death or exceed the rate of deterioration. They go into log. They increase dramatically. Stationary is the rate of growth is equal to the rate of death. Okay, so they stay like this. Okay, this is 500, 10 grow and 10 die. Still 500, 500, 500. You call that stationary. If the death exceeded, you call that dead. So lag, log, stationary, dead. There is something called flow cytometer, and this is something cytometer counting the cells. Cytometer counting the cells. They determine the size and they differentiate. This machine can differentiate between dead and live cells. The most simple of that is the turbidometry, and this is another way to see these viable bacteria. Okay. To analyze the population, you need to see the colony count, and you count the cells. If you're using this plate, which is called the viable plate counting, you are going to count the colony and the cells. if you're taking that sample, what each one of those represent? This, I'm computing. Each one of these spots is a colony that represents a type of cell. Okay, this is a summary of what we already talked about. How they get the nutrients, the transport, how they adapt, the partnership, and expanding the population. And that's it for this chapter.